All right, this is the podcast for genetics. All right, so what is genetics? It is the study of heredity. And heredity is one of our properties of life that we talked about on the first week of school. So heredity's definition is indented a little bit. It is the transmission of traits from parent to offspring. So it's passing on those genes, those traits, that DNA from generation to generation. All right. So I don't know if you know who this guy is, but he is the father of genetics. He is the one that figured all this genetic stuff out. And his name is Gregor Mendel. He lived in the 1800s. You don't need to know his dates or anything, but in the 1800s is when he was around. And he liked plants. And you can see that in the picture of this drawing that someone did. And he wanted to be a plants teacher, actually. But the rumor is that he couldn't pass the teacher exam. He took it a couple times. And so he ended up becoming a monk in Austria, is where he lived. And he still worked with plants. And he did a lot of experiments in his little monastery. So why was Mendel successful and not other scientists? Because there were other scientists in the 1800s that were also working on these types of experiments. But Mendel was successful for two reasons. Number one, he used a garden pea. So if you think of a pea, you know, that vegetable that you eat or like or might not like, that's the plant that he used that got him to be successful. And so on your outline, there's 6 reasons why the garden pea was great as his test subject. Number one, they're small. I mean, he would live in a monastery. He didn't have a lot of space. So he needed something small that he could work with. Number two, they're easy to grow. It didn't take a long time for him to get them to grow. Number three, they produce many offspring. So think science experiments. He's getting a lot of data. So as he was crossing these peas, he would get a lot of offspring, which meant he had a lot of data to work with and analyze. Fourth reason why he chose the garden pea is they mature quickly. He didn't have to wait around a long time to see the results. Fifth reason why he chose the pea, there's many varieties of it. It's not just a green little pea that you eat for dinner. There's actually many varieties of the pea plant. And so I'll show you that diagram in a minute. And the last reason why he chose the pea is that they're easy to fertilize. So you're thinking genetics, right? Heredity, traits going from parent to offspring. He needed to fertilize that pea. And the thing about a pea is that you can self-fertilize it, or in a plant's case, you can self-pollinate it, which means you can take the sperm from one pea and put it into the same sperm's eggs on the same plant. Or he could cross-fertilize. He can take the sperm from one pea and put it into the egg of another pea. Again, plants, they're not necessarily called sperm and egg, but you get the point. All right, so that's why he chose the pea, and that was the first reason why he was successful. The second reason why he was successful is he used a quantitative approach. He didn't just say, ooh, look, I have green peas and yellow peas. He said, ooh, I have 527 green peas and 164 yellow peas. He used numbers. Going back to one of the reasons why he chose the pea, here are all the different, well, this isn't all of them, this is a some of them, varieties of the pea. So you can see that seed form. They can be round or they can be actually wrinkled. They can have different color seeds, yellow and green. In their pod, because they grow in a pod, the pod can be inflated or it can be restricted. It can be green or yellow. And then if you look at the flowers can be axial or terminal. Axial means it's like along the stem in the middle. Terminal means it's up at the top. The seed coat color can be different. It can grow tall or short. And there's more. So there are a lot of different traits that he can examine with the garden pea. All right, so let's look at his experiment. It is a famous experiment that you'll have to know and understand the 3 steps to it. Step number 1, he had to produce a parent generation. So these are the first groups of peas that he's going to cross. And so we're going to call that parent generation the pea generation. So he's going to allow these pea plants to self-fertilize for many generations. He wanted to make sure that they were pure, like true bred. So that means that we're going to be talking about purple and white. He wanted to make sure that the purple peas, these are flowers, only produce purple peas, and that the white peas only produce white peas. They are completely purebred. Okay. So what he did is he took one pure purple pea plant and one pure white plant was his pea generation. So that's what he is starting with. Okay. Step number 2, he's going to produce the first generation. So he's got a pure purple and a pure white pea plant. The first generation is going to be called F1. So what does he do? He's going to cross-fertilize this purple and this white pea. So he could have took the white sperm and put it into the purple's eggs, or he put the purple sperm and put it in the white eggs. He's cross-fertilizing them, okay? And so what was his results? You would think that he would have maybe like, remember he used numbers, 50 purple, 50 white. You know, what did he get? It actually wasn't. And to his surprise, he got all offspring for the first generation were all purple. Even though he had a white pea plant and his parents, right, for his parents, he didn't have any white offspring. So we thought that was super weird. Like, where did the white trait go? So obviously he's going to take this one step further. Step number 3, he's going to produce the second generation. So like the grandkids, right? And so this is the F2 generation. You can't think of F1 as first generation. Like here, F actually stands for filial, which means son or daughter. So step 3, he produces the second generation, which is going to be called the F2. He's going to take 2 of those purple plants from F1, that, you know, the kids, and he's going to self-fertilize them. All right. So now what were his results? Did any of the white generation come back? Were they still all purple, 50-50? And the white did come back. 75% of the offspring were purple, 25% of the offspring were white. And it didn't matter what trait he was looking at. If he was looking at tall and short, it was always a 3-to-1 ratio, no matter what trait he used. So here's a picture visualization of his experiment. His pea generation, he had a purple plant and a white plant that were self-fertilized and purebreds. He cross-fertilized them and all of his F1 flowers were purple. Then he self-fertilized these purple plants and he ended up with a 3-to-1 ratio of 3 purple to 1 white. And it didn't matter what trait he was looking at, he always got that 3-to-1 ratio. All right, so Mendel came up with some conclusions. He figured out and basically proved heredity. He proved that parents can transmit information about their traits to their offspring. He proved heredity is true. He also figured out that each individual pea plant, and now we can refer this to humans as well, each individual person has 2 factors for each trait. Factors, I mean, that's a weird word. He didn't know what a gene was back then. This was back in the 1800s. They didn't know what a gene was. They didn't know a lot about DNA or anything about that yet. So he knew that though, that every individual has 2 factors for every trait. You get 1 factor from your mom and you get 1 factor from your dad. So he figured that out. These factors or genes that eventually we figured out that they were genes are represented by letters. And so we're going to have letters to represent these genes. These letters are also called alleles. You're going to see this word a lot. So you need to understand what an allele is. All right, so an alleles. If both alleles are the same, meaning the same, you get the same letter from your mom and you get the same letter from your dad, then you're going to be considered homozygous for that trait. Homozygous means the letters are the same, or the alleles are the same. If the alleles are different, the one you got from your mom and your dad are different, then you are considered to be heterozygous for that trait. So if you know your prefixes, that helps you out. Homo means the same, hetero means different. Okay, those are the first 2 words. There's like 6 terms that you really need to understand genetics. And this is kind of like math because we're going to be building on this and doing genetic problems. So you really need to understand those first 2 terms. All right, next 2 terms. Traits can be described in 2 different ways, a genotype and a phenotype. When I ask you in a genetic problem for a genotype, I'm asking you for the alleles or the letters that represent the trait. Every trait can have 3 genotypes. They can be big P, big P, little p, or little p. It doesn't matter what letter you're using. Usually, well, we'll talk about the letters situation in a little bit, but those are the only 3 options. They can both be capital, they can have one of each, and they can both be lowercase. When you have one of each, one capital and one lowercase, the capital letter is always written first. Okay, so these are the letters, these are the alleles that represent the trait. One of them you get from your mom, and the other letter you get from your dad. Also, when you do genetic problems, I'm going to be asking you for phenotype. Phenotype is the expression of the trait. What do you look like? And so for our example, that would be purple or white. Okay, so that's 4 terms right now. Last 2 terms you need to understand, and you probably have heard these words before: dominant and recessive. All right, so dominant is the capital letter. So when you saw those genotypes and you saw the big P and the big P, little p, those capital letters, those capital P's, those are dominant letters, dominant alleles. To be dominant, you only need 1 capital letter to express that trait. So a big P plant would express the big P trait, whatever that letter is standing for. Even if you have big P, little p, you are still going to express the dominant trait because the recessive is going to be masked. You're not going to visualize it. You're not going to see it or express it. The only way you will represent and show a recessive trait is if you have both lowercase letters. All right, so that might be a little bit confusing, but you'll get it. All right, so let's double check and see that you understand. So there's a little chart, or there's a space for the chart. We're going to talk about 4 different columns. Genotype, you need to understand what a genotype is. When I give you a genotype, you should be able to tell me is it dominant or recessive. When you look at the genotype, you should also be able to tell me if it's homozygous or heterozygous. And when you look at the genotype, you should be able to tell me what the phenotype would be if you knew what it stood for. All right, so we have 3 genotypes possible for our pea plants. Pea plants can be big P, little p, or little p. Okay, those are the only 3 possibilities. All right, so just looking at the big P, is that dominant or recessive? Well, I see capital letters here, so that is definitely dominant. Okay, homozygous or heterozygous, those P's are the same, so that is homozygous. And in the example that we did with purple and white, if you had a big P genotype, you would end up having purple flowers because purple was dominant in Mendel's experiments. All right, next genotype, hopefully you're understanding this. Big P, little p, dominant or recessive. I see a capital P, doesn't matter that you have this here. That capital P kind of trumps it, if you want to say that word, and that is going to make it be dominant. Homozygous or heterozygous, those letters are different. One is capital, one is lowercase, so that is heterozygous. And just because even it has that capital P, it automatically is going to express the purple flower. Even though it has the gene or the allele that represents the white, it's not going to express it because that dominant trumps it. All right. And then lowercase p is going to be recessive because there's no capital letter there. Those letters are the same. That's going to be homozygous. And that is the only way, if you have both lowercase letters, it's the only way that you will express a recessive trait. All right. If you do not understand this chart, you need to email me, text me, something, get with a friend. This is very basics and you have to understand this. Okay. All right. A couple more things to finish up the podcast. I want to show you this picture. These are chromosomes. And so I just wanted to show you that these letters, these alleles are actually on the chromosomes. And so if this was mom's chromosome and this is dad's chromosome, you can see that the letters are in the same location. They're called loci. You don't need to know that term. It's an AP biology term, but I'm just showing you that the letters are there in the same exact spot on the chromosomes. Okay. All right. So Mendel came up with 2 laws. Mendel's law of segregation. Segregation. You should know that word. Segregation means separation, separating. So here's the actual law, terminology of the law. Members of each pair of alleles separate when the gametes are formed. A gamete will receive one allele or the other. There's a typo, or the other. This occurs in meiosis. So let me explain. Members of each pair of alleles separate when the gametes are formed. So let's say this flower is big R, little r, right here, right? Big R, little r. When this flower is going through meiosis and it's creating its 4 sperm, let's say, which are pollen, the big R is going to go to one pollen or sperm, and the little r is going to go to a different sperm. Those letters separate. The big R, little r does not end up in the same sperm. They separate. So one sperm only has one of those letters. Okay. Same thing with the eggs. The gamete, the sex cell, the sperm or the egg, whatever way you want to talk about it, is only going to receive one letter. Okay. That's his first law. He figured out. His second law is the principle of independent assortment. That one states that 2 or more pairs of chromosomes separate independently of one another during the formation of gametes. This is random. So basically what that means is that, let's say you have 2 different traits, all the capital letters aren't going to end up in the same sperm. And all the little lowercase recessive letters, alleles, aren't going to end up in one sperm. Everything is random when things are forming. Okay. All right, last thing is, why did Mendel's results repeat? And that is due to probability. So we're going to be doing genetic problems. We can predict now what offspring can look like if we know the genotypes of the parents that we're doing a cross for. So before we get into that, it's a different whole note section. I want to talk about probability a little bit, and you should be able to predict and be able to do ratios, okay, or percents. So we're going to talk about coins first, something basic. What's a chance of flipping a head on a coin? We're going to look at cards. What's a chance of getting a diamond on a deck of cards? What's a chance of getting a 9? What's a chance or the probability of getting a 9 of diamonds? And then we're going to look at the sex of children. What's a chance of having a boy? What's a chance of having a girl after having 4 boys in a row? All right, so let's just go through each one of these and make sure you understand ratios and probability. So for a coin, there's 2 sides on a coin, right? The chance of flipping a head, well, there's only one head out of 2 sides. So that is 1 out of 2, one chance out of 2 possible outcomes. That's how the ratio is written. Or you can put that in percent, it would be 50%. Okay, you need to be able to do both of those. Cards, what's the chance of getting a diamond? So you'd have to think, how many diamonds are in a deck of cards? How many cards are in a deck? And if you got that right, you would have 13 diamonds in a deck of cards out of 52, which actually, if you knew that 13 times 4 is 52, then you would know that it is a 1-to-4 ratio, or 25%. You would have to divide that out to know that. All right, 9 of diamonds, or 9 by itself. What's the chance of getting a 9? There's 4 9s in the deck, right? So 4 out of 52, which does reduce to 1 out of 13. We are not going to be putting that into a decimal percent. We're just going to leave it as 1 out of 13. The 9 of diamonds is obviously just one of those, 1 out of 52. We're not going to divide it out and give me a percent in decimal form. I know you guys like math, but we're not doing that. All right. The chance of having a boy child, well, you can have a boy or a girl, so it's 1 out of 2 or 50%. And a chance, the chance of having a girl after having 4 boys in a row, it does not matter. It is still 1 out of 2 or 50%. All right. And that finishes our podcast.